Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the basics of quorum sensing in bacterial cells. And so one of the first questions we need to ask ourselves is what actually is quorum sensing? And in very basic terms, it is a way in which cells communicate with one another, much like how we might communicate with one another. However, to be more specific, it's a cell density dependent communication system that allows bacterial cells to synchronize and coordinate gene expression using chemical signals. Now, the chemical signals used to communicate between each other can be small molecules or proteins that the bacteria secrete into their local environment. Now, when such molecules are used for cellular communication, they are referred to as autoinducers. And so when we take all of this information together, bacterial cells produce signals in the form of autoinducers, and when the concentration of the autoinducer reaches a critical threshold, the cells will collectively respond to the signal, facilitating a coordinated population-wide change in gene expression. Now, if we were to visualize this on a graph, plotting autoinducer concentration on the y-axis, so the amount of signal being produced, and population concentration on the x-axis, relating to the number of bacterial cells, we can see that at low cell densities, bacteria begin to produce autoinducer molecules. But since the population is sparse, the concentration of these signals remains relatively low. At this stage, individual cells behave independently, carrying out basic metabolic activity and survival functions without coordinated group activity. However, when the bacterial population increases, autoinducer molecules begin to accumulate in the environment, and so when a critical threshold is reached, which is known as a quorum, the bacterial cells shift collectively as a population, and they initiate various group behaviours by altering gene expression. Now, in the context of medical microbiology, this will usually result in behaviours such as biofilm formation and the production of various virulence factors. And so at this stage, individual cells are acting as part of a collective whole rather than in isolation. Now, the relationship between signal concentration and population density is the essence of quorum sensing, and it essentially allows bacteria to behave as multicellular-like communities when their numbers are sufficient. But one of the key things we need to consider is why. Why might it be in a cell's best interest to turn on or off select genes at high cell densities? Well, there are several reasons. First, it allows bacteria to optimize resource use. You see, processes like biofilm formation and virulence factor production require significant energy, and so by only activating these genes at high cell densities, bacteria can launch a coordinated and high-impact attack on the host, increasing the likelihood of overwhelming the host's defense systems. Second, it helps bacteria evade host defenses. So if virulence factors were expressed at low cell densities, they might trigger an immune response, while the bacterial population is still too small to withstand it. And so by delaying activation until a critical mass is reached, bacteria can remain undetected until their numbers are large enough to pose a significant threat to the host. And finally, quorum sensing enables spatial coordination. Essentially, it informs bacterial cells about colonized and uncolonized areas of the host, allowing some cells to switch phenotypes and spread the infection beyond the initial infection site, helping to establish a more extensive infection. Now, ultimately, quorum sensing is a highly effective strategy that allows bacteria to act as a collective force, maximizing their chances of survival and success within a host environment. Okay, so now that we understand the significance of quorum sensing, let's break it down a little further. All bacterial quorum sensing systems rely on three core principles that enable bacteria to communicate and coordinate their behaviour. Now, the first is production. This is where bacteria continuously produce and release autoinducers into their surroundings. The second is detection. Once autoinducers reach a critical threshold, receptor proteins detect them. And finally, we have reaction. Upon detection, a response is triggered within the cell, altering gene expression and turning on various group behaviours. Now, a key feature of all quorum sensing systems is that they facilitate a positive feedback loop. You see, once bacteria detect a threshold level of autoinducers, they don't just react, they amplify the process by producing even more autoinducers, which increases detection rates and leads to an amplified reaction, creating that positive feedback loop. And so on the plus side, this allows bacteria to transition very quickly from individual to group behaviour, making quorum sensing an incredibly efficient and dynamic communication system. However, because all bacterial quorum sensing systems are based on the same key principles, it does throw up some problems from time to time. You see, when multiple bacteria or even different strains of the same species coexist in the same environment, they each produce their own autoinducers, which may be similar, yet slightly different. And so rather than activating their own quorum sensing system independently, these signals can cross-inhibit each other, disrupting communication. Now, this cross-inhibition can occur through several general mechanisms. The first revolves around competitive inhibition, structural mimicry, and receptor antagonism. This is essentially where autoinducers from different species or strains compete for the same cell surface receptor. In some cases, an autoinducer might bind to a receptor of a different species or strain, but fail to induce the necessary conformational change for the signal transduction to occur. This effectively blocks the correct autoinducer from binding and triggering its intended response, preventing proper quorum sensing activation. The second is signal degradation. This is where some bacteria produce enzymes that degrade or chemically modify the autoinducers of competing bacterial species or strains. And so by breaking down these signal molecules, they can disrupt the communication networks of their rivals, gaining a competitive advantage during colonization of a new host. Now, quorum sensing systems do vary between bacterial groups. Gram-positive bacteria rely on a system that uses auto-inducing peptides. These are small modified proteins that are transported out of the cell and detected by membrane-bound receptors. In contrast, gram-negative bacteria use acyl-homoserine lactones, and these are small diffusible molecules that freely pass through the cell membrane and accumulate in the environment, diffusing back into the cytoplasm at high concentrations and interact with cytoplasmic receptors. Now, whilst both have the same three basic principles, their molecular mechanisms differ, reflecting the unique cell envelope structure of the gram-negative and gram-positive bacteria, as well as their respective autoinducers. However, there is a third system. This is found in both our gram-positive and gram-negative bacteria, and it's known as the autoinducer 2-like system. Now, as autoinducer 2 is found throughout the gram-positive and gram-negative bacteria, this effectively makes it a common language for interspecies communication. And this is different from the autoinducing peptides of gram-positives and acyl-homoserine lactones of gram-negatives, which act as an intraspecies communication system, essentially meaning it is species-specific. And so because autoinducer 2 facilitates cross-species communication, it plays a crucial role in complex bacterial communities, such as those found in the gut microbiome or in polymicrobial infections. Okay, for the last bit of this video, we're just going to briefly look at an overview of our two main systems, starting with our gram-positive system, which is based upon the production of auto-inducing peptides. So very briefly, quorum sensing begins with the production of an auto-inducing peptide within the cytoplasm. This can occur either directly through translation or via an auto-inducer synthetase enzyme. In both cases, the peptide is initially produced in an inactive form known as a pro-auto-inducing peptide. And so to activate the signal molecule, it must first be transported out of the cell. and during this process, a membrane-associated enzyme will modify the peptide, converting it into its functional autoinducing form before releasing it into the external environment. Now, as bacterial cell density increases, these autoinducing peptides gradually accumulate in the external environment, and once a critical threshold is reached, the autoinducing peptides will bind to a membrane-associated histidine kinase, triggering autophosphorylation. Now, this phosphate group is then transferred to a response regulator inside the cell, leading to changes in gene expression. Now, as is common of all quorum sensing systems, there is a positive feedback loop where the response regulator upregulates both autoinducing peptide production and its own signaling components, ensuring a rapid and synchronized response across the bacterial population. And finally, we have the gram-negative quorum sensing system, which is based on the production of acyl-homoserine lactones. So again, very briefly, quorum sensing begins with the production of acyl-homoserine lactones by an autoinducer synthetase enzyme within the cytoplasm. Now, unlike the peptide-based signals in gram-positive bacteria, acyl-homoserine lactones are freely diffusible, meaning they pass easily across the cell envelope and accumulate both inside the cell cytoplasm and in the external environment. Now, as bacterial cell densities increase, the concentration of acyl-homoserine lactones both inside and outside the cell gradually builds up. Once a critical threshold is reached, acyl-homoserine lactone molecules will bind to their corresponding autoinducer receptor, inducing a conformational change that activates the receptor's transcription factor activity. This leads to changes in gene expression, and again, just as we saw with the gram-positive system, there is typically a positive feedback loop where the response regulator upregulates both acyl-homoserine lactone production and its own signaling components, ensuring a rapid and synchronized response across the bacterial population. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.